All right, so it's my pleasure to welcome Ryan Abernathey to our ESE seminar. Ryan is an associate professor at Columbia University. He got his PhD from MIT and did a short postdoc at Scripps before joining the faculty at Columbia. In his work, he's been seeking to understand how ocean eddies and their transport of heat, carbon, and other tracers affect the large-scale structure and circulation of the ocean and how the ocean responds to climate forcing. He's also been leading the community in developing better software tools to analyze and visualize large data sets. And we had originally planned for him to visit in person in the spring, which would have been more fun. But we're very happy that we've been able to spend the day with him, calling in from Vermont, where he's on sabbatical. So Ryan, it's a pleasure to have you, and the floor is yours.
Great. Thanks a lot. I really appreciate the invitation, and I enjoyed meeting with everyone today. So I recently had the chance to contribute a chapter to a book on ocean mixing on the topic of isopycnal mixing. And that really sort of got me thinking about some of the big picture questions that I want to share with you today. So this is going to be a very kind of broad overview talk, and it's a new talk that I have not given before. So I appreciate your patience as I work through the inevitable kinks. Let's see, important message to open my talk. I think it's just important to acknowledge that it's great to that we're carrying on and having seminar series and doing our best to keep science going. But it's also important to remember that this is still a really challenging time for a lot of people. There's so much going on in the world right now. I personally find it very hard to concentrate at times on science when it feels like the end of the world is nigh on several dimensions. So I don't know who needs to hear this, but I think it's important to state. And in terms of my own situation, there's a significant chance that I will be interrupted by a 4-year-old who is not used to having his dad in the home office at this time of day. So I will attempt to handle that with grace if it does happen. So the basic outline of the talk, I'm going to spend quite a bit of time, probably too much time, talking about some fundamentals of isopycnal mixing. But I'm assuming that this audience, like my own department, is a very broad one that doesn't necessarily think about ocean mixing and transport all day. So I'll try to introduce the problem from a fairly general viewpoint. There's going to be kind of two main parts. I'm going to talk about some of our work we've done studying the impact of isopycnal mixing has on ocean tracers and water masses using low resolution numerical simulations, where we basically use isopycnal. We just have a knob that tells us how strong isopycnal mixing is, and we turn that knob and we see what it does to the ocean. It's a simple but interesting approach. And then I'll switch gears and talk about how we are trying to use very high resolution simulations that directly resolve the mixing process to learn more about isopycnal mixing. And I'll conclude with some thoughts about where we need to go in the future for isopycnal mixing research. So just to set the stage very generally, the ocean is a stratified fluid, and it's stratified in density, and both temperature and salinity contribute to that stratification. And so here we're looking at some sections across the Atlantic that show the sort of the warm, salty lens of the thermocline that overlies cooler, fresher water. And so it's very important to recognize that the stratification is not uniform in space, but it actually varies. And that creates tilt to these density surfaces, which we'll call isopycnals. And this creates a very important anisotropy in ocean motion. And so I want to spend some time introducing the idea of the neutral plane. So imagine we have these contours. We're sort of looking at a cross section of the ocean. These are like, think of them as density surfaces with light water overlaying dense water. And if we take a water parcel and we perturb it upwards, it's going to be denser than its environment, and it's going to experience a buoyant restoring force pulling it back down. And the same thing will happen if we push it downward. It's going to be restored back to its original position by gravity. And that will lead to some oscillations. Forgive my crude keynote animation there. And those oscillations happen at a frequency called the buoyancy frequency. You don't have to, understand these equations, but if you're an oceanographer, this is what we call the buoyancy frequency, or Brunt-Väisälä frequency, n squared. That's proportional to the stratification in temperature and salinity. And it sort of sets this stratification sort of sets the frequency of internal waves. But another point is that if we perturb that same parcel horizontally, we'll also experience a restoring force in the vertical. So it will try to move back up to its level of neutral buoyancy. And really there's a special direction along which we can perturb a water parcel where it won't experience any restoring forces. And that direction is what we call the neutral plane. And mathematically, we can quantify it as just the direction that is perpendicular, the plane that is perpendicular to the local gradient of potential density. And so the point is that there's this special direction in the ocean interior whereon water parcels can slide around without experiencing any restoring forces. And a key point is that this neutral motion doesn't change potential energy. And so, in writing this review chapter, I realized that this point is actually really important because so much of ocean mixing research is focused on the energetics of mixing. The energy inputs needed for mixing this and that. And in this sense, isopycnal mixing is essentially invisible to the energy budget. It's not directly constrained by energetics. And so it's kind of lives in its own world, and we need a different framework to think about it and understand it. Now, a technical point, but an important one that I wish someone had told me when I first got into this field. This neutral plane is pretty well defined, locally at any point in the ocean. But it's a pretty big jump then to go from that to what I call global neutral surfaces. So the point is that because the ocean's equation of state is nonlinear, there is not actually some density variable that we can plot whose isosurfaces or isopycnals are parallel to this neutral plane everywhere. And this makes it difficult to analyze ocean transport because we don't actually have a perfectly well posed coordinate for the isopycnal direction. And a lot of people, there's a lot of work to try and get around this, and oceanographers use something called neutral density, which is kind of like a best approximation at that, but it's still not a perfect one. And so I'm going to continue to kind of ignore this problem in most of the rest of my talk, but it is a real issue and should always be kept in mind whenever we start talking about isopycnal surfaces and isopycnal coordinates. So what is isopycnal mixing about? What's the phenomenology of isopycnal mixing? So I've hopefully convinced you that there's this special plane that water parcels can slide around without any energy. And then all we need to get isopycnal mixing is some stirring mechanism, something that is going to jostle those water parcels and cause them to slide around. And the dominant mechanism for that is mesoscale turbulence. And so these are some visualizations of what we're looking at here is just a slice across the Southern Ocean. This is from a model. Shows the depth of a particular isopycnal surface. So we're looking at temperature. And so what we can see like the basic phenomenology of isopycnal mixing in this. There are these eddies and filaments on scales of 50, 100 kilometers that are stirring up the temperature along this isopycnal surface. And that stirring motion is working to kind of homogenize tracers along the isopycnal plane. And indeed, we do observe that a lot of tracers are uniform along isopycnals for this reason. But more generally, most tracers exist in some sort of force dissipated equilibrium where large scale forcing sort of tries to regenerate these gradients, and then isopycnal mixing and other mixing processes work to eliminate them. A great framework for thinking about isopycnal mixing can be found in this thickness weighted average framework of Bill Young. So this is an analytical framework that we can use to study stratified fluids. And I'm going to spend a little bit of time talking about some elements of it. So the way we construct this tracer budget in this thickness weighted framework is that we average the ocean over many realizations of these eddies, and we construct this thickness weighted average tracer concentration equation, which looks kind of like an advection diffusion equation. The term on the first term, d sharp c dt, is a material derivative, and then but it involves the advection by a residual velocity, and then we have this other term, this divergence of some eddy tracer flux, J. And the mathematics here are somewhat, complicated, and the notation is somewhat ugly, but there's an important point that comes out of this, which is that those eddies that we saw on the previous slide show up in two places. On one hand, they show up in the advection, and that's sometimes what we call eddy Bolish transport. So it's fundamentally an advective effect of eddies. And they also show up in the eddy tracer flux. That's this J term. And when our fluid is what we call adiabatic, when there are no real major sources or sinks of density in the ocean interior, this term here goes away, this vertical or diapycnal flux term, and we see that the eddy tracer flux lies in the isopycnal plane. Okay, so from there, it's a relatively short step to a very common framework to thinking about isopycnal mixing, which is that we can derive a variance equation that states that that says that on average the flux of this tracer along isopycnals has to be down the main gradient, down the background gradient of the tracer. That's kind of a fundamental process of many, or a fundamental aspect of many mixing processes. And here we can kind of see sort of from first principles that it predicts that tracers will be fluxed along isopycnals down their background gradient. And so that leads to this really central concept in the way we think about isopycnal mixing, which is trying to represent this isopycnal mixing process through some diffusion coefficient, K. Okay, so this diffusion coefficient really kind of measures the strength of isopycnal mixing. I could give a whole talk on K, but that's not really what the direction I want to go in. Another important aspect is that isopycnal mixing is really only a well-defined concept when we're in a part of the ocean where density or buoyancy is conserved to leading order. When density is not really conserved and there are processes actively changing the density, then that constraint that the eddy flux of tracers along the isopycnal plane it's not required anymore, and we're free to flux tracers in any direction we want. And so the way we normally think about this is that in the so-called adiabatic interior of the ocean, where things are mostly conservative, we have isopycnal mixing, and then up by the surface, the same mixing process becomes more or less horizontal, mixing tracers sort of in the horizontal plane. But how exactly we transition between those two layers is a pretty open question. And we can diagnose this from simulations. I'm going to skip that. Okay, this is a horrible slide, so I don't ask you to read it. This is literally just copying something from our paper about this, but all I really want you to read are the bold words here. So we kind of identify what we see are some of the real challenges in quantifying isopycnal mixing. Inhomogeneity means the fact that these mixing processes vary strongly in space and time. It's not as if we can represent isopycnal mixing by one number, although actually that is what many climate models try to do. Isopycnal mixing is highly anisotropic, so mixing can act very differently in different directions, and this is often tied to the orientation of the potential vorticity gradients. Mixing is highly non-local. So by that I mean, or at least in many places, it's likely non-local, that the stirring and the dissipation don't necessarily happen in the same place. And that assumption is fundamental to a lot of frameworks for both observing and theorizing about mixing, but it's no reason to believe it's a good assumption to make. And finally, we know that mixing varies a lot depending on scale. So, whether we're talking about mixing at the 100-kilometer scale or the 10-kilometer scale, the strength and processes could be very different. And so the consequence of this is that observing isopycnal mixing is very hard. It's a really difficult observational problem to actually constrain, design a field experiment and actually constrain and measure mixing. So direct methods for measuring isopycnal mixing usually involve putting something in the ocean, either floats or some kind of tracer dye that you actually squirt in the ocean. There are also indirect methods that use mixing length theory, that use surface observations, or use inverse methods to get at isopycnal mixing. But those involve a lot of assumptions, and so are therefore sort of fundamentally less useful than those direct methods. So my career as an observational oceanographer began and ended in 2009 when I participated in this field experiment called the Diapycnal and Isopycnal mixing experiment in the Southern Ocean. This was a huge, international, multi-institution field campaign. Maybe some of you were involved. That deployed a lot of floats and tracers, and it lasted many years and cost millions and millions of dollars. And we learned a lot. It was a great experiment. But in terms of isopycnal mixing, this is basically what we learned from that campaign. So really from those floats and from those tracers, we were able to get kind of one number out of the ocean of how strong isopycnal mixing was. And of course, there's a lot of good science behind this and a lot of useful stuff that happened. But a lot of what was useful involved numerical models, and showing that numerical models would give you the same number in this spot, and then sort of extrapolating from there and seeing what else the models had to say about isopycnal mixing. So these field campaigns are very important, and they're very valuable for studying isopycnal mixing. But at the same time, we just don't have the resources or the money to sort of scale up this approach and get these direct measurements of isopycnal mixing everywhere. So the bottom line is that there remain large uncertainties in the magnitude and spatial pattern of isopycnal mixing. I showed you one number. I could review some other studies that argue that isopycnal mixing in the ocean, its strength should be somewhere between 10 and 10,000 meters squared per second, right? So different methods for inferring isopycnal mixing and measuring isopycnal mixing give you numbers that span several orders of magnitude. So given this situation of a lot of uncertainty, and really sparse observations, our approach has been to use numerical simulations to try and learn more about isopycnal mixing. And that's what I'm going to spend the rest of my time telling you about. And there's sort of two directions, as I said at the beginning. One, we're going to use our system models and ocean models and examine the impacts of varying this K parameter, which describes the strength of isopycnal mixing, over a wide range, and see what it does. And then on the other hand, we're going to use some high resolution simulations, which we have reason to believe can represent the isopycnal mixing process fairly well to directly study isopycnal mixing and the associated transports. So first, I'll start with this, this impacts of variant K. So I sort of got into this through some collaborations with Anand Ganadesikan, and I'll just briefly mention two studies that we worked on. One was to ask how ocean CO2 uptake depends on K, this isopycnal diffusivity parameter. And so for this, we used a fairly coarse resolution global Earth system model, the GFDL ESM2MC. And in this model, the default is that isopycnal mixing is just parameterized with a constant diffusivity rate. And so we ran a bunch of experiments with different values of isopycnal that are broadly, isopycnal mixing that are broadly within the observed range, as well as some other more sort of spatially variable estimates that I helped produce. And we looked at the total amount of CO2 that the ocean takes up, which you see in the figure on the right, and then the flux of CO2 into the ocean under these different conditions, which you see on the left. And the bottom line is that isopycnal mixing can, make a considerable difference in how much CO2 the ocean takes up. A difference on the same order of magnitude as, different policies we could take to mitigate, carbon emissions, right? And so getting it right would be really useful. And the other thing I want to just draw your attention to from this is that the Southern Ocean is where things matter most for isopycnal mixing. And that's because the Southern Ocean is a place where we have isopycnals outcropping and essentially provide a window for stuff from the surface to mix down isopycnally into the ocean interior. And so changing isopycnal mixing can double or cut in half the rate of CO2 flux into the Southern Ocean. Pretty simple point. Just another study that I'll point out is that we looked at how changing isopycnal mixing influences El Nino. It's not something I had ever even thought would happen, but it turns out that a stronger isopycnal mixing gives you more ENSO variability, stronger ENSO variability. This was counterintuitive to me at first, but I think for people who study ENSO, it's not. Really, the main impact that the way this works is that changing isopycnal mixing changes the thermocline structure in the tropical Pacific. And what's really most important for ENSO is the background state and the strength of the vertical stratification in the tropical Pacific. So by increasing isopycnal mixing, you actually weaken the vertical stratification in the tropical Pacific, and that eliminates a damping of ENSO that can happen due to that strong thermocline in the eastern tropical Pacific. So the point here is just to say, actually, isopycnal mixing is somewhat neglected, parameter in our system models, but it does matter for things we care about. So
hey, Ryan,
Ryan. Yes, Andy.
I'm just letting you know, you have a question in the chat as to whether changing the magnitude of isopycnal mixing is related to diapycnal mixing, or whether they're independent.
I would argue that to first order, they're independent for the reason I stated at the beginning, which is that, diapycnal mixing, the way we think about diapycnal mixing is it's so wrapped up in energetics. We have these strong energetic constraints. You need potential energy to change, to do diapycnal mixing. We constrain our parameterizations for diapycnal mixing based on the energy budget and all that stuff. And none of that stuff really applies to isopycnal mixing. I mean, it depends on mesoscale. It's not to say there aren't feedbacks and relationships between them, but to first order, I don't think they need to have any relationship. Thanks for the interruption. Yeah, I can't see that chat, so
yeah, exactly. We'll keep an eye on it. So actually, that kind of gets to my question, though, right? That I agree with you. I think in terms of that the isopycnal mixing is somewhat invisible to the energy budget, but then of course the degree to which you can stir is some function of the kinetic energy in the ocean, right?
Absolutely.
So I don't know. I guess I don't understand really well how those pieces all fit together. And of course, those are open areas that people are still working on. But
sure. So like you could imagine that we could change the diapycnal mixing, and that would change the strength of the eddies, and that would change the strength of the isopycnal mixing. This morning, Jörn told me some work they're doing looking at sort of mesoscale internal wave interactions, where the internal waves can damp the eddies, and that changes the eddy transports. I'm also reminded of David Marshall's work on bottom drag. Changing bottom drag affects isopycnal mixing as well, because it changes the eddy energy. So anything you do that would change the mesoscale field can influence the strength of isopycnal mixing, and probably will. Okay, so one thing about all these experiments is that we're changing K, but the circulation, that the MOC, or the overturning circulations, is also changing, right? It does depend on K, and so there's some feedbacks in there. So it's somewhat hard to attribute directly certain things we observe in this type of simulation to changes in isopycnal mixing. So with postdoc Spencer Jones, we set up some idealized experiments that wanted to get really much more into detail on how exactly the role isopycnal mixing is playing in setting the distribution of water masses in the ocean, in the global ocean. And to do this, we ran these experiments with some idealized ocean models that use this kind of, to me, I call this double Drake geometry. So this is basically a large basin meant to represent the Pacific, a small basin meant to represent the Atlantic, some very idealized surface forcing. And the reason we use such idealized experiments is, one, they're simple to understand, and two, we need to be able to run them for a very long time. Because what we did is we forced tracers at the surface. Each of these colors in this figure is a different tracer that we forced to basically to the value of 1 in that box and 0 everywhere else. And then we run these experiments out to equilibrium. It takes 5,000 or 10,000 years. And what happens is the ocean fills up with tracers whose value all add up to 1 at every point in the ocean, but those relative ratios tell you where that water originated from. And so there's a really long-standing question, actually, that we got into from talking with Wally Broker at Lamont before he passed away last year. One of the last problems Wally was working on was how much Atlantic water is in the deep Pacific, right? It's kind of this really first-order fundamental question. And these type of experiments can help get at that question. So here's what the overturning circulation looks like in these simulations. It's reasonably realistic in the sense that we have an AMOC in the Atlantic, and in the Pacific, we have kind of this deep clockwise overturning cell. And the experiments we did, we left this circulation totally fixed and only changed the isopycnal mixing as it was applied to tracers. And we looked at how this water mass distribution depended on the isopycnal mixing coefficient. And we think these are interesting because so much of our reasoning about tracer distributions is tied to the MOC, the overturning circulation. And so here's an experiment where we don't change the MOC at all, and we just look at what mixing can do to water mass distributions. So here's sort of the main result. What we're looking at over on the left is the concentration of Atlantic water in the Pacific. I seem to have removed the color bar, but basically this varies between 0 and 1, right? It's a fraction. What we see is for very weak isopycnal mixing, there's a lot of Atlantic water in the Pacific. And we can almost completely get rid of all of that by making isopycnal mixing stronger. And what you can see over on the right is a sort of just bulk characterization of those water mass origins as a function of mixing parameters. The first three are changing isopycnal mixing. I don't think I have a pointer, unfortunately. That would be nice. But you can see the same thing that you see in the other figure, which is that as you increase isopycnal mixing, the fraction of Atlantic water in the Pacific really goes down. And so why is this? Again, I put another equation on my screen, which probably shouldn't have put there because I'm not going to really explain all the terms in it. But what we've done to understand this is we've developed a one-dimensional advective diffusive model that can reproduce the results in these simulations pretty well. And the basic mechanism is the following. When you have very strong isopycnal mixing, the isopycnals outcropping in the Atlantic Ocean that are carrying North Atlantic origin water to the Southern Ocean, those get diluted very strongly with Southern Ocean origin water. So it's what really matters is the concentration of Atlantic water that is in the ACC, how much water of North Atlantic origin that is carrying. And that really depends strongly on isopycnal mixing. So since doing this first paper, Spencer has a new paper where he compares a sort of modern day configuration with an LGM configuration. And
Ryan, real quick, there's a question of clarification. So Dimitri is asking if you force your conditions to 1 in the basins, shouldn't you at steady state just get a uniform tracer field? There must be something else that you're doing.
If we only use one tracer, that would definitely be the case. But there's like 12 different tracers. And so if you add them all together, you absolutely do get exactly what Dimitri is talking about. You just would fill up the whole ocean with this tracer. However, each tracer has its own different origin. So that allows us to trace the surface origin of the water at any point in the simulation. Hope that answers the question.
Yeah, and so there's another restoring to 0 elsewhere for
exactly.
each tracer? Yeah.
The tracers are restored to 1 in their region, but they're restored to 0 everywhere else. So the tracers are destroyed as well. They're not just created. And that's very important for, I mean, I think that matches how we think about water mass origin. If there's an outcrop, we don't call that North Atlantic water anymore. Once it's reventilated, its origin is erased. And that's what this sort of forcing accomplishes.
And just not to be too pedantic, Ryan, this is Jess, but they're restored to 0 everywhere else in the surface. In the interior, it's whatever the circulation is doing to carry them around.
In the interior, they're completely conservative. They're advected and diffused following the model circulation.
Yeah.
So yeah, it's a one way to study water mass origins. It assumes a steady state. We can also look at things like age and transit time distribution, but this is the method we chose because we think we understand it. It's pretty simple actually at the end of the day.
Great, thanks.
So I'm gonna kind of move on to the second part of the talk because I can already see I'm behind time. I'll have to make some tough decisions about what to skip. So the bottom line from these studies, they've been interesting to us. We convinced ourselves more that isopycnal mixing kind of really matters for global scale ocean tracers and water masses. I didn't even mention cabling, but it's part of the story as well. And so, it's fun to turn this knob of isopycnal mixing and see what happens. But what we really want is a better understanding of isopycnal mixing such that we can parameterize it, with great confidence in an ocean model or a climate model, and feel confident that even if we don't directly resolve eddies, that isopycnal mixing will be represented correctly. That's sort of the holy grail of isopycnal mixing research, at least the way I see it. It's a parameterization problem. So the way we've been studying that is with high resolution channel simulations. So by channel simulations, I mean these sort of boxes where we can simulate the ocean at very high resolution And channel, I mean, they're sort of periodic in the X dimensions, kind of like the ACC. Southern Ocean is probably the most important place for isopycnal mixing because of these steep isopycnals and outcropping. And so we have this sort of toy setup, this laboratory that we've been using for a lot of different studies. And if you've seen me talk before, I've been doing this for my whole career, working with this type of simulation. One thing though that we can do now that we couldn't do 10 years ago is run, these like 2,000 by 2,000 kilometer simulations at 1 kilometer resolution, pretty fine resolution. You start to see some new and interesting stuff start to come out. We've done a couple different things with isopycnal mixing in this type of configuration. I'm going to show you a movie from some work by Dhruv Balvada, where we looked at tracer uptake, the role of eddies play in taking up a surface tracer. And what you're going to see is just now here, we're just going to be squirting a tracer in at the surface and watching how its concentration evolves. And on the left, we'll see that concentration at 250 meters depth at a constant depth surface. And then on the right, we'll see the same tracer interpolated onto the 3-degree isotherm, which is, in this model, we only have temperature. So it's the same as an isopycnal. And so let's just watch the movie and see what happens. So this simulation never equilibrates. We're just keep pushing more tracer into the ocean. And so we can see it being taken up here. And we can see, over on the left, we have these really fine, filamentary structures. And I'll maybe play that movie again. Over on the right, we see the same thing from the isopycnal perspective. And actually what's striking to me is how different these two things look, right? So when we look at the tracer on an isopycnal, we really see that its concentration is very strong near the outcrop. That's what that blue line is, by the way. That's the outcrop. It's kind of mixed into the interior by these filaments and these eddies. So this is just to give you kind of flavor of what these simulations are like and what they do. Dhruv's paper is really cool, but for the sake of time, I'm not going to talk about it. Instead, I'm going to talk about something that has a kind of a little bit broader Earth system interest, which is the way we've used this setup to study the role of eddy transport and isopycnal mixing on the phytoplankton ecosystem in the Southern Ocean. So a really important property of the Southern Ocean in terms of phytoplankton is that phytoplankton growth is strongly limited by the availability by light and then the availability of iron. And iron has a couple different sources in the Earth system. There's certainly source of iron from the surface, but there's also a lot of iron available at depth. And so the way that, our understanding of the sort of seasonality in the phytoplankton ecosystem and the strong bloom that we observe in November, December, January, is that the bloom has the seasonal cycle because, of course, light, but also mixed layer depth changes. And we're suggesting that there may also be a role for mesoscale and submesoscale turbulence in supplying this iron from depth to feed the phytoplankton. Just to get a little bit more detailed on that, here's a really nice figure from a paper by Tayebue that shows the sort of seasonal cycle of the phytoplankton ecosystem and the transition between different phytoplankton types throughout the year. And the basic idea that they propose is that, during the winter, there's no light, but the mixed layer is very deep. And so that deep mixed layer brings a lot of iron into the mixed layer. And then over spring, sort of the mixed layer shoals, but the iron is still there, and the phytoplankton can eat it throughout the summer. And a big question, they argue in this paper that there has to be a lot of recycling of iron throughout the summer and the fall to sustain the observed productivity, because they say there's no other real source of iron into the ecosystem at that point while the mixed layer is shallow. So our hypothesis that we wanted to check out with these simulations is whether eddy transport could help bring iron into the mixed layer through another mechanism, a three-dimensional mechanism that wasn't considered in this paper. So again, we've got the same type of simulations, but this time we embed a biogeochemical model in it. And I'll let this play while I talk about this model. So we have, again, this sort of high resolution yet idealized physics, and then we couple this to a biogeochemical model based on the Darwin model, but simpler. It has 2 phytoplankton species and 1 zooplankton. It models phosphate, nitrate, and dissolved and ligand-bound iron. So, intermediate complexity biogeochemical model. So what you can see here is basically this color is a phytoplankton biomass. You can see this sort of seasonal cycle. We have a seasonal cycle, by the way, in this simulation of, I'll play it again, of phytoplankton. We have a bloom, and we also clearly have a signature of eddies that are advecting this phytoplankton around and also helping to transport iron. So we tuned our simulation and we provided an iron source at the northern boundary such that we basically reproduce within the error bars, the vertical profile of iron as observed by the GeoTraces experiment. And so you can see from this figure that we have enhanced iron at depth and then low iron at the surface due to biological consumption. So this is what the seasonal cycle of biology looks like in this model. So we have a strong phytoplankton bloom. And so these figures kind of show an average seasonal cycle as a function of depth and then the month on the x-axis. So and kind of averaged over the center of the domain here. So that green blob is a phytoplankton bloom. And with that comes a big reduction in iron concentration. And that black dashed line is the mixed layer. So the mixed layer depth, which is changing significantly also over the seasonal cycle. And what we're looking at now on the bottom is the vertical transport of iron, split into 2 contributions. One from which is the vertical eddy transport, and then the vertical diffusive flux. So that's from the mixed layer scheme in the boundary. And so what you can see here is that vertical transport by the boundary layer scheme is very strong, but it's confined, of course, completely to the mixed layer. Whereas the eddy flux is playing a really important role in providing iron across the mixed layer base. We can also see that even during summer, there's a continuous supply of iron due to eddy fluxes at the mixed layer base, which kind of makes us question whether we need to invoke the mechanism of recycling to sustain production, if there's always new iron being injected into the euphotic zone. So we've ran these simulations at different resolutions as a kind of way of probing the role of eddies. And you can see as we move from 2 kilometer to 5 kilometer to 20 kilometer resolution, the strength of that vertical transport decays significantly. And on this figure, what we can show is that primary production and total iron transport really strongly go up with resolution. They go up together. So the eddy resolving simulations are the red dot, the red points on this graph. And on the y-axis, you see the total phytoplankton biomass, and the x-axis, you see the eddy iron flux. And so the productivity goes up at high resolution, even though the mixed layer is shallower. And so we're really convinced that this is due to strong dependence. It's really due to this sort of predominant role that eddy transports play in supplying iron to the phytoplankton ecosystem. Let me just skip that and go right to this. So as a final step, we have a lot more sort of detailed turbulence style analysis of the iron fluxes, which I'm not going to go into here. I'll just show this one figure that is in wave number space. So on the y-axis, we see the depth. And now on the right axis, we've broken up this vertical ion transport spectrally using a wave number Fourier cross spectrum. And so we can really see what scales are actually contributing to this transport. And on the left, you see the buoyancy flux, and on the right, you see the ion flux. And so it's interesting to see that pretty small scale processes at scales down 10 kilometers and smaller are making a really significant contribution to this vertical ion flux. And those are happening pretty deep, 300, 400 meters depth. So I think there's a lot to work out still here to understand the physics of this transport and get to the point where we can parameterize it accurately in ocean models. Just to kind of close this part, can this flux of iron be parameterized? So this is kind of now coming back to isopycnal mixing. So I mentioned that eddy transports can be thought of as both having an advective and diffusive part. The advective part we associate with a parameterization called Gent-McWilliams, and the diffusive part with the component we call ready. And so we did a lot of tuning, and we found that we could make a coarse resolution model with a 100-kilometer resolution, basically reproduce these vertical fluxes and their seasonal cycle. It took a lot of tuning and a lot of playing around with different parameterizations. Some things we learned from that exercise is that this is really not mixed layer instability, at least it's not represented by the mixed layer instability parameterization, the Fox-Kemper et al. Parameterization. That is active only in the mixed layer, and in order to get this, we need transport below the mixed layer. Sort of the traditional Gent-McWilliams and ready parameterization can produce these transports, but you need a couple special ingredients. You need appropriate spatial variability, and so we use the Visbeck scheme to get that, and you need the right vertical structure. In particular, we found this Ferrari et al. Parameterization, which thinks about the diapycnal part of the eddy flux near the boundaries, which I mentioned earlier in my talk, is really important for getting this iron into the mixed layer properly. And then finally, by just turning the ready part on and off, we concluded that isopycnal mixing is at least 50% of this iron transport, at least from the framework of the parameterization. This again points to the need to properly represent isopycnal mixing in our coarse resolution models. In this case, if we want to get the biology right and all the associated impacts on the carbon system. Should mention that the work I just talked about was all done by my former student Takayo Uchida, and is available in these papers you can see here, who is now in Grenoble. So just to wrap things up, where do I think we should go in the future for isopycnal mixing research? Well, obviously, I really think we can learn, we can learn a lot from models about isopycnal mixing, much more than we already have. I think we can, that if you look at the observational studies like DIMES, what they show is that our high resolution numerical models can match the observations very well, and that they can resolve the isopycnal mixing process. So as a tool for studying the physics of isopycnal mixing, models are the way to go. And that's very different from diapycnal mixing, at least on a global scale. We can't hope to really reasonably model, internal wave breaking on a global scale and the associated energy fluxes. But I think we can with the latest generation of global eddy resolving ocean models. So this is a model that probably many of you are familiar with. It's these MIT GCM LLC 4320 simulations, global 1 to 2 kilometer mesh run by folks at JPL, including Dimitris. And these models are absolutely fascinating in terms of the number of processes and that they resolve. And I think by studying isopycnal mixing in models like this, we can get around, go past a lot of the limitations we see in our idealized channel simulations. We can confront some of these issues of anisotropy, of inhomogeneity, and use them as laboratories to understand isopycnal mixing better and eventually quantitatively validate parameterizations for isopycnal mixing, and perhaps even develop data-driven parameterizations, incorporating new tools from machine learning that can really learn the proper parameters for isopycnal mixing directly from data. Something we'd love to do. If you know me and if you've talked to me recently, you know that I'm kind of obsessed with the technical challenge of doing this. You know, when I first saw this simulation, which came out about 5 years ago, I was flabbergasted, and I thought we were going to discover so many new things from it, and to some degree we have. On the other hand, we're really hampered by the difficulty of working with this type of data. So this simulation produced over 1 petabyte of data. You're not going to just download that to your laptop and, you know, make some quick calculations. And so, you know, the belief that these type of simulations are really valuable to us has really kind of changed the direction of my career recently. So I've been working very hard on this project that we call Pangeo, which is aimed at really building the software tools and the infrastructure we need to work with this scale of data and have it still be fun. By that, I mean, still be able to get answers on a somewhat interactive timescale rather than having to wait for, you know, weeks or months to chew through data. This project basically involves taking challenging scientific use cases like this and finding sort of the open source software that we think is a promising start to solving those challenges and contributing to that software, making it better, helping it integrate, and then deploying it in high performance computing and cloud computing environments where we have both the data and the computing to work with satellite data and model data at the petabyte scale. And we've been at this for a few years, and it's been pretty satisfying, and I think we're making a lot of progress. And so if you feel this pain as well, I invite you to check it out. With particular relevance to isopycnal mixing, we work on this Python package we call XGCM, which is an open source package for GCM analytics. And it can do things like, you know, evaluate budgets, energy budgets, and stuff like that. And we also recently added the capability to do vertical coordinate transformations, like transforming from depth to isopycnal coordinates, which is, you know, kind of this first step for studying isopycnal mixing and transport. Yet it's often a real bottleneck just in terms of the analysis. And this package is really designed to be able to scale out. So if you have a cluster, you can use it on a cluster. If you have access to cloud computing or GPUs, you can have these tools work on GPUs. And so what I hope is that, you know, if I come back and talk to you in a few years, I'll be able to tell you how we've come full circle and use these tools and this infrastructure we've been working on through this Pangeo project to really make some new discoveries about isopycnal mixing from these high resolution simulations. So that's where I hope to go. I don't really have a conclusion side, but I'll just end by saying thanks. If you want to find out more about my research and some of the papers I mentioned, you can find those on my website, which is listed here. And if you want to find out more about our Pangeo project, you can find that on the Pangeo website. So thanks a lot for your time.
All right, thanks so much, Ryan. Clap, clap, clap. I'm gonna